Okay, guys, give me a pathway that has intrinsic kinase activity. Well, that's so the kinase, the MAPKK is not necessarily a pathway. I mean, it's involved in many different pathways, but that was more used as an example of intracellular signaling molecules that help to relay the message. So there's no receptor there, right? Huh? Yes. NF kappa B or SMADs? Okay. Let's see. Nope. The question was, would NF kappa B be an example of a nuclear receptor? Why and why not? Here, let me just quickly. Yep. Okay, so we got an example. We've got an example of intrinsic kinase activity. That would be SMADs, right? And that's TGF-beta signaling. Thank you. JAK-STAT was an example of kinase associated. Notch is an example of signaling through a receptor on another cell, right? Where you've got regulated proteolysis, so we'll get back to that. Wnt beta-catenin is an example of GPCR. What else? Okay, so I got a question about NF kappa B. I believe NF kappa B is not, but I feel like I need to double check more closely because I was using that as an example of signaling through regulated proteolysis of an inhibitor to activate a latent gene regulatory protein, which you shall get to. Okay, so let's move forward. Okay, so we have an example of intrinsic kinase activity, which are SMADs. We have an example of kinase associated, which is JAK-STAT. Okay, so let's talk for a little bit about relaying the signal. So we've got all these different receptors. We've got examples of different pathways for the different receptors. Let me put this here. This is notch for this one. Notch for the receptor on another cell. And then you guys should all be thinking about CD4, right? Because we've done CD4 in recitation. Okay. So you get activation of a pathway. And then what? When you're talking about, so like we've got the nice one with GPCRs and adenal cyclase and CAMP, and this isn't all using these second messengers, right? Small, I think the book called it small intracellular mediators. But what happens, so what else do we have going on? So let's say you want to activate, we talked about RAS, okay? So where does RAS come in? Right, so does anyone remember what RAS is? It's a molecular switch, which means what? It turns on and off. How? GTP, GGP, gaps and gaps. Okay, good. So let's just move forward from here. And I don't want you to think RAS is only associated with enzyme coupled. I believe there's also with GPCRs. So we'll include that in this. Oops. So what do we know about RAS? We know it's a GTP binding protein. We know that it turns things on and off. What? It's in the membrane. So we know it's in the membrane, so it's close to the receptor. And so now we're talking about, so we've got all these different types of receptors, and they relay messages to the nucleus. How does RAS relay? Yes?
I just had a question. I thought in your institution when you told me that it could be recruited, it's not always there.
Did I tell you RAS? Okay.
RAS gap and gap.
There you go.
Okay.
Right, so RAS is membrane associated, but its regulation is based upon its gaps and gaps, right? Gap activity and gap activity, right? So, what's the job of RAS though? What does it do in the signal transduction pathway? Does anyone remember? Because it's great that it's a GPCR and all that. Yes?
Doesn't it relay signals from the cell surface?
So it relays signals from the cell surface. But what else does it do? Yes?
It's other kinases, phosphorylase.
It activates other kinases that phosphorylate gene regulatory proteins. What else does it do? These are all right. Yes?
It promotes cell proliferation.
It promotes cell proliferation. Well, yes. So in the end, it activates all this stuff, gene expression, promotes cell proliferation. But within a signaling pathway, it also spreads the signal from one receptor to multiple pathways. So RAS can act to activate multiple signal transduction pathways from one receptor. So RAS is going to spread the signal. It's a G protein. It's regulated by gaps and gaps, right? But how so? If it's in the membrane, it's regulated by gaps and gaps. How does it get turned on from signaling? So you have signaling molecule binds to your RTK, and then eventually RAS will be activated. How? Phosphorylation of what? I know, that's always the answer. Phosphorylation of something, yes.
Isn't like the G protein not phosphorylated, and it recruits like it recruits another protein that recruits that?
Okay, so something, not the G protein, something's phosphorylated that recruits the RAS gap. That's a receptor, right? Your receptor. So remember, you've got your little picture of your cell membrane and your receptors, and they get phosphorylated, and RAS is hanging out here. That was a really ugly membrane, but it's probably more realistic. RAS is hanging out here, but when ligand binds, you get phosphorylation of receptor, and you get recruitment of some adapter proteins, which then recruits the RAS geth, which activates RAS, right? It's getting small. So that turns on RAS, which helps to relay the signal from the plasma membrane to nucleus. It helps to activate gene expression. It helps to activate multiple pathways from one receptor. Yeah.
Can you repeat what you just said?
So you got your receptor. Here's your. It looks like a person. So you've got your. There's your ligand. There's your receptor and your ligand. That's your plasma membrane. RAS is associated with your plasma membrane. Ligand binding results in activation of receptors. Remember, they've got kinases, either associated or intrinsic. Phosphorylation of the cytoplasmic tail results in recruitment of the RAS-GAF through usually recruitment of an adaptive protein. But then RAS-GAF is there. RAS-GAF can kick off GDP, add GTP, turn on RAS, and then RAS can do its thing. Okay? What other molecules did we talk about that were involved in signaling? That might be activated by RAS?
Is that intrinsic? Yes, is that intrinsic or is that
I think RAS can be activated by many things. So we can double check it.
the phosphorylation of the receptor?
For that, just think of phosphorylation. Don't think about why it could be intrinsic. But it's really about phosphorylation and recruitment of the gap. Okay. What else? What else helps to relay? So now we're talking. So we've got all our receptors. We're talking about relaying our signal to the nucleus. We talked about RAS. What else did we talk about? Important. Importance are very important for getting transcription factors into the nucleus. But let's step back a bit. Yes. MAP kinases, because you got to get. So here, like, think of this as the top of the cell over there. We got to make our way to the nucleus. It looks like our nucleus appears to be starting. Well, it's over there, and it's over here too. Okay, so we got to make it to the nucleus. So you've got our MAP kinases, and the MAP kinases it's a module of 3 different kinases that phosphorylate each other and activate each other and help to pass the message, right? So there's MAP kinase, let's see, kinase, MAP kinase. And they're involved in a lot of different pathways, and the cells are really crowded place. So how do we keep them together and stop them from interacting with all these other proteins? Scaffold proteins, right? So MAP kinases kind of held together as a signaling molecule by a scaffold protein. Oh my God, I'm dying here. Please tell me there's more chalk somewhere here. Okay. Sorry, that's going to be loud. Okay, so you've got your scaffold proteins. What do you think? Do you think I'm doing a good job?
Yes.
Okay. Looks like craziness up here to me. Okay, so scaffold proteins are holding your MAP kinases together, and that's still relaying the signal. And so then what? You get phosphorylation of things, right? Because these are all kinases. And some might be other signaling molecules, some might be transcription factors. There's a lot of things that are activated through MAP kinases. Okay? Am I missing anything here? Anyone wishing I mentioned something? I'm sure I'm leaving all sorts of things out. Yeah? Huh?
Did you say how it gets turned on from signaling?
How what gets turned off from signaling? How does RAS get turned off? Turned on?
Yeah, I was gonna say.
Yeah. So signaling, so let's go back. RAS, signaling results in phosphorylation of your cytoplasmic tail, which recruits your RAS-GAF, directly or indirectly. Okay. So then you get, this results in phosphorylation of some sort of protein that might end up going. So this is like a very general mechanism. Something gets phosphorylated. MAP kinases don't have one target, or MAP kinases have one target as many targets. So you might phosphorylate a transcriptional regulator that will then go to the nucleus. Yes.
So are the MAP kinases just like there, or portions of the RAS have to be activated in order for the MAP kinases?
Those are two different mechanisms which may work together or may not, depending on the pathway. Okay. And so that's why there was that one figure in the textbook that showed GPCRs and kinase-associated receptors activating multiple pathways. And so, and then, like, there might be some consistent things up top, but then it's just like black boxes kind of to the bottom. Okay, so what else? Where do we go from here? So then we got phosphorylation of some activators, proteins. And that affects activity. Activity might be going into the nucleus and activating gene expression, might be activating something else. Okay. Do they affect latent gene regulatory proteins? I guess they can. So let's go to latent gene regulatory proteins. Okay, so we have all these intracellular signaling molecules that we're talking about, and their purpose, remember we started outside with signaling molecules, and it's receptors, and now we're kind of in the middle of the cell with all these signaling molecules, and then eventually you get activation of some kind of gene regulatory protein. And so that brings us to the concept of latent gene regulatory proteins, and that just means it's a protein that's present and kept inactive until signaling makes it active in some way. And there were a bunch of examples of that, and I'm losing space. So, oh dear, I'm running out of chalk. This is horrible. That one? This box is pencils. It's horrible. Oh, wait, there's chalk here too. It's all bad news over here. Okay. Okay, so let's talk about latent gene regulatory proteins, because that's one thing that might end up getting phosphorylated and affected in some way, right? So then you have, oh my God. Latent gene regulatory proteins. Okay, so I gave you lots of examples of latent gene regulatory proteins, and they were meant to just be examples of different mechanisms of activating or inactivating, right? So who can throw out a latent gene regulatory protein? JAK-STAT. How is that latent, and how is it activated?
You said that it's latent because it's inactive.
So it's, I guess, so it wasn't the two, I don't even think I talked about JAK-STAT when speaking of latent gene regulatory proteins, but it does fall into the category because you have your stats there, but they're not active until they dimerize and go into the nucleus, right? So it does fall into that category if you're thinking of something that's inactive and then changes in some way and becomes active, right? Does everyone agree with me? Yes. No? Yes. Okay, so let's write. So we have, so let's not call it JAK-STAT, let's call it by the name, let's call it STAT, because that's the true protein. NF-kappa B. So STAT needs phosphorylation and then dimerization. And then it can go into the nucleus. Okay, NF-kappa B. How is that kept inactive? Right, so it's bound to an inhibitor. NF-kappa. And so it's normally, so IKB inhibitor, right? Kind of redundant. Oh, yeah! You get an A+. Did you have to steal it from another room, or did you go to the
I
office?
took it from my one of my teachers' room. She doesn't even know it's there. It's like a big box of chalk. She always looks at chalk.
Now watch me break this. Yay! Okay, NF kappa B. So you've got I kappa B inhibitor bound to NF kappa B that keeps it in the out of the nucleus by covering up its NLS, right? What happens to it? What happens? Well, how does this latent gene regulatory protein become activated? Or what's degraded in the proteasome? I kappa B is degraded. How is it degraded? How is it identified to be degraded? It's phosphorylated. So, like, you have the signaling through receptors that activates kinases that phosphorylates I kappa B, and then you get degradation in the proteasome. Okay. Yay. Okay, so STAT, NF kappa B, what else is a latent gene regulatory protein? Beta-catenin. Beta-catenin. That's kind of different, right? So how's it latent and inactive? It's being made, it's constantly being transcribed and translated, but it's constantly being degraded. Right. So how is it being degraded? Right, GSK3 phosphorylates it. So again, we have some signaling molecule that exists in a way to phosphorylate something, but it's kind of the opposite here. So that's why I always say be careful with phosphorylation. It just depends on what's what. So beta-catenin is normally phosphorylated by GSK3 and degraded. And so how do we activate it? Yeah. Right, so then we prevent degradation of beta-catenin. So active by stabilizing. Right, by sequestration of GSK3, because GSK3 is the kinase that's phosphorylating it so it can be degraded, right? Okay, give me another one. Another latent gene regulatory protein. SMAD. Okay, so SMAD. So how are SMADs inactive? Conformation, right? So based on their conformation, they're inactive. And when you get phosphorylation of one of the SMADs, that results in interaction with the other one, and you get this oligomerization, which results in it going into the nucleus, right? So do you have anything being degraded here? No. So, but I had a lot of questions about latent gene regulatory proteins. Are they always being degraded? No. So you can think of like the heading being latent gene regulatory proteins, and then there could be two mechanisms, one involving cleavage or proteolysis, and one involving change in conformation or dimerization or something like that, right? So SMADs, that's how I should have organized it, right? Oh well. So phosphorylation, oligomerization, nucleus. Okay, so let's see. I'm going to put a degradation. I'm going to write like I put a big D for degradation in front of the ones that require degradation, right? So that's degradation. These are not. Okay, so there's one more. What other one did we talk about? You guys are doing great. Huh? Wnt, we did that, beta-catenin. Yep. What else? Notch, right? Notch and delta. So notch So notch, remember that's the one where it's telling the cell nearby not to follow in its path, kind of gene expression. So notch is on the cell, and then delta is on the other cell. So it's an example of binding to a receptor on another cell. So notch, how does that work? How is it kept latent? Yeah. It's stuck in the membrane. So this was a receptor that gets cleaved at some point, remember? So the receptor cytoplasmic tail gets cleaved after binding to delta. So it's in the membrane. Wait, in membrane. Receptor cytoplasmic tail is cleaved after okay, I'm running out of room now. After binding to delta. So what could you say about the NLS on notch? Just if you had to guess. Is there an NLS? Well, so remember, it gets cleaved. When it gets cleaved, what does it do?
It makes the second cleave on.
Okay, so it gets cleaved. There's multiple cleavages. When you have your final cleavage, what happens? Where does it go? It goes into the nucleus and changes the repressor into an activator to activate gene expression. So based with that, what I tell you, does it happen in NLS? Yes, it has to. And what's keeping it out of the nucleus is that it's normally physically kept out of the nucleus by being in the plasma membrane. And you can probably think about all of these in terms of NLSs and how those NLSs are covered up or not available until they need to be, right? That'd be a beautiful essay question, right? For like a final exam. So like stat, remember you have to dimerize to create the NLS and get in. NF kappa B, the NLS is covered up by I kappa B, beta catenin. It has an NLS on it, but it's being degraded, so it doesn't make a difference until it's stable, and then it can go in. SMADs need to dimerize for an conformational change from dimerization that allow NLS to be available, and then notch, the NLS is there, but it's stuck in the plasma membrane, so it can't be used until it's released. Okay. So in the last 15 minutes, we'll talk about turning off. So remember, so this is like a lot of what we did over the past few weeks, right? But we need to think about when you have signaling and turn on, it needs to see, why do we spend weeks here when I can do it in one lecture, right? We stop? We just stop going to lecture, right? We just like come for one lecture, and we'll do this. And then this is actually how graduate school is. You sit there for an hour and they spew it all at you, and then the next meeting they do it again. But it's like, oh, it's horrible. I mean, it's fabulous. I learned a lot. Let's talk about turning off signaling. Okay. So some people visited me earlier today, and I said, "Quick, how do you turn off signaling? Tell me all the mechanisms." And I think they came up with it slowly but surely. So let's do a quick turning off signaling up here. Okay. Someone give me a mechanism for turning off signaling. Endocytosis. So receptor downregulation through endocytosis. Okay, what else? Give me another one. So I heard degradation. So you can get endocytosis and then degradation via the lysosome. So downregulation, endocytosis, and that could be degradation. What else can be degraded? that's going to turn off signaling. A ligand? What else can be degraded? Inhibitors, does that turn off signaling? We're talking about turning off signaling. If you degrade an inhibitor, you're going to turn on signaling. What? A transcription factor. So anything, any of the things between the receptor, well, between outside the cell all the way to your transcription factor, you degrade any of those things, you're going to turn off signaling, right? So we talked about degrading JAK. We talked in the JAK-STAT pathway. We talked about your degrading receptors through endocytosis, which, by the way, downregulation endocytosis could be through multiple mechanisms, right? You can get what we talked about in the last unit, where you'll get monoubiquitination and degradation, endocytosis, the degradation of a receptor, but also we talked about the arrestin phosphorylation of GPCRs, right? So this is in the same category, but more specific to GPCRs. And we talked about it over here somewhere. Right? And that acts as both for endocytosis and inhibiting interaction with the G protein. What else? GPCRs, degrading rate. So degrading, since this is also receptors. I'm going to keep going higher. So then also degradation of any signaling molecule. Okay. What else? Block the cytoplasmic tail through what? Okay, so you can get phosphorylation in recruitment of inhibitors. And what's the opposite of that? What's the opposite of phosphorylation? Right, dephosphorylation through the activity of phosphatases, right. Right, so phosphatase, if you remove those phosphate groups that were creating those docking sites to recruit, let's say for RAS, right? You can turn off RAS through activating a phosphatase. And, if you upregulate a phosphatase or introduce a phosphate group on the cytoplasmic tail, then you can't recruit the RAS gap anymore. And you're not going to be able to activate RAS anymore. And then at the same time, you turn off RAS for the gap. Did I forget anything? I'm sure I did. Did I get the bulk of it down? Did this help in organizing things?
Yes.
Yes, it's very organized.
It's crazy land, right?
I want somebody to drive by me who's walking right now.
Yeah.
It's
probably worse in an ortho class, right? Where it's
Yeah, it's most shapes, it's all shapes.
Okay, well, if you don't have any other questions, that'd be that.